Thank you for joining me today to listen to my investigation of the translational frameshift within EA bacteriophage tail assembly chaperones. So to begin, tail assembly chaperones are present in both contractile and non-contractile double-stranded phages and are highly conserved. There are two isoforms of this protein that are essential for tail assembly, a short isoform as well as a frameshifted isoform. During tail formation, both of these isoforms bind via its N-terminus to the tape measure protein to stabilize and prevent it from precipitating out before the rest of the tail has been assembled. The frameshifted protein, however, has an additional role, which is to bring the major tail protein into close proximity to the tape measure protein so that it is able to polymerize the tail. Once this polymerization begins, the tail assembly chaperones dissociate and are not part of the mature tail. So the program translational frameshift that I will be focusing on today has been highly researched within bacteriophage lambda. About 96% of the time, the ribosomes translate the open reading frame of gene G to make that one product. About 4% of the time, however, though, the ribosomes translate the open reading frame of gene G. Once they reach the slippery sequence, slips back a nucleotide, which then forces the ribosomes to start translating the reading frame of gene T to form this fusion protein, or also known as a frameshifted protein. And note how gene T is not one of the products made. So the four bacteriophages that are under study for my investigation are Winsescapine, Strathy, and teddy bear, which belong to the EA1 subcluster, as well as quartz that belongs to the EA10 subcluster. The University of Ottawa's phage hunters have annotated the genomes of Winsescapine and quartz. During the annotation process, we are unable to call the translational frameshift within the tail assembly chaperone genes. This is due to the fact that the slippery sequence is not evident, as it does not follow the conventional pattern, and the type of frameshift that would arise from the slippery sequence is not typical to the most commonly called -1 frameshift. If this frameshift did occur, we would be able to annotate it, as did APA from the subcluster GA. We were told we would need to provide experimental evidence to show that this frameshift does occur, which led me to my hypothesis that the EA phages do undergo a translational frameshift within their tail assembly chaperone genes. For me to test my hypothesis, my first objective was to determine if this fusion isoform can be expressed in a heterologous E. coli system. So firstly, we amplified the tail assembly chaperone fusion isoform via PCR. We inserted these fragments into vectors so that they could be tagged with GST. We transformed these plasmids into E. coli. We grew a few of these colonies and induced them with IPTG and collected these samples so that we could run a protein gel. Kamasi stained protein gel of two different samples, uninduced and induced. We have the empty GST plasmid as well as the plasmid with the Winsescapine fusion isoform insert. So when these samples are induced, we are seeing a prominent band that could be representative of the short isoform. We also have this band that could be representative of the frameshifted protein, but we aren't confident to make that conclusion quite yet. We also ran this with Strathy and quartz GST tagged isoforms, and we are seeing the same results. So this led to our second objective, was to raise polyclonal antibodies against this frameshifted isoform. So in order to do this, we amplified two fragments of the C-terminus of the frameshifted protein. We had a small fragment, which is 100 nucleotides, as well as a larger fragment of 300 nucleotides. We then cloned them the same way, tagged them with GST, transformed them into bacteria, induced them and collected samples, and then purified these samples via affinity chromatography. Once we eluted the protein, we then ran a protein gel and stained it with Kamasi so that we could visualize the two fragments. We then injected these fragments into rabbits to raise the polyclonal antibodies. The small fragment injections occurred twice, while the large fragment injections occurred 3 times, and each injection occurred 4 weeks in between each other. Once we collected the sera, we were able to test it on our Western blots. So here we have the same samples with our empty GST plasmid as well as the GST plasmid with the WINS insert, and we have been able to see these two isoforms present. And note that we're using the sera, we have not purified the antibody, so there are a lot of GST antibodies within the sera, and that's why both of these isoforms are being detected. So here we can say pretty well that the fusion protein is occurring as well as the short. However, to be more confident in our results, we wanted to construct a different plasmid with a different tag so that we could only see the frameshifted protein. So here we used an MBP tagged fusion isoform, and as you can see, only one band is being detected, so we're more confident in our results that the frameshifted protein is apparent. We also carried out the same results with our Strathy and quartz constructions, and you can see the same results are present. This led to our third objective, is that we wanted to see when the tail assembly chaperones were produced when an infection occurred with its host bacteria in vivo. So we added high titer phage lysates to the host Mycobacterium foliorum at 100 multiplicity of infection. We then collected the samples at 10 different time points and ran our Western blots against our antibody. So here are our results. we can see that there's difference in the timing of the tail assembly chaperone production as well as the abundance even within the same cluster. And it's also important to see the differences between the EA1 cluster as well as the EA10 cluster. Some future directions for this investigation would be test the antibody on other phage clusters with annotated tail assembly chaperone frameshifts to confirm them, as well as to identify differences between clusters and subclusters. Most importantly, to identify and determine the consensus slippery sequence within the EA bacteriophage cluster. This can be done using mass spectrometry as well as site-directed mutagenesis. And finally, to create an antibody to the short tail assembly chaperone isoform. So in conclusion, EA1 and EA10 bacteriophages produce the frameshifted tail assembly chaperone protein. Our raised antibody is able to detect the frameshifted protein and can be used to analyze differences among bacteriophage clusters and subclusters. And thirdly, tail assembly chaperone expression may depend on infection timing, and abundance differs among phages within the same subcluster. I'd like to acknowledge and give my thanks to all of these institutions that make the C-Phages program possible and all the listed people. Thank you for listening to my presentation today.